Please join me in welcoming my colleague, Irina Mueller, who is also from the Allen Institute on the assay development team, and she will be giving us a talk called Cell Organization During Human iPS Cell Mitosis.
Thank you, Caroline. 1 year later, we also don't have more lines. We also have new and exciting data, and I'm really excited today to be able to share a glimpse in the dividing mitotic stem cell with you. So once we have our endogenously tagged stem cell lines made and fully validated, the microscopy team images these lines on an automated standardized imaging platforms and thousands of replicates. And all cells are stained with dyes to visualize the plasma membrane and the DNA to serve as cellular reference structures. All those cells are being segmented, and all good segmented cells are being uploaded into the Allen Cell Explorer. The 3D cell viewer in the Explorer contains currently 20,000 segmented cells. Unfortunately, I don't have time today to actually go and show you really the 3D cell viewer, but I highly encourage us to go on our website and play with the Explorer and go through our cells in 3Ds and spin them around. One thing that the assay development team is doing is actually gets to explore the data, and so can you. So you can go to our website, allencell.org, go to Tools and Data, and then click at this 3D cell viewer. Today, we're just going to look at single cells of our mitochondrial tagged line. And even if we just look at the image gallery and not even go into more detail in the cell, I hope you can appreciate the large variation we see in cell morphology, where the nucleus is, where our structure is that we tagged. So one of the questions that came up is, where does all this variation actually come from? And one potential glimpse to an answer is when we actually start going into our interactive plotting tool, where you can plot multiple features against each other. Today, we're going to plot the cellular volume against the nuclear volume. And when we do this and look at all our tag lines, we can actually see two populations emerging. There's a larger population, and when we look at those cells in there, they are all non-dividing cells. However, if we go into the smaller, lower populations, all of the cells in there seem to be dividing cells. So we wanted to understand the physical organization of the mitotic cells. To do so, we sorted all our images into 8 cell stages. And those cell stages were solely based on DNA characteristics. But when we used images from our tubulin tagged cell lines and sorted it into the cell stage categories, we actually can see that they match nicely biological relevant cell stages. We have one interphase stage, so non-mitotic cells. We have 2 stages of prophase, 2 stages of prometaphase, 1 metaphase stage, 1 anaphase stage, and a combined stage of telophase and cytokinesis. We have a manual data set of approximately 9,000 cells, of which 700 are mitotic. And we're really excited about this data set because having endogenously tagged gene-edited stem cells allows us to understand the organization of a diploid non-transformed cells without the need of overexpressing proteins. We also have currently 14 major organelles and key intracellular structures that we looked at. So we can, instead of looking at one structure at a time, hopefully look at all those major structures in an integrated fashion. Now that we have the data set, we can address the question how cell morphology and structure morphology and localization changes as the cells go through the cell cycle. Let's look at the cell morphology first, since we saw that huge variety of cell shapes when we looked at the cell viewer. This is a movie of a stem cell colony, and you can see a dividing cell. And as the cell divides, the cell rounds up and rises up within the colony. So we wanted to quantify the morphology. First, we looked at cell roundness, and we quantified cell roundness as the shortest axis of the cell, which is the longer one, divided by the longest axis of the cell. And the closer that ratio to 1, the rounder the cell. So if you look at the cells as they are in our 8 stages of cell cycle, we can see that around prometaphase or cell cycle stage 3, we see the cells to round up, and they continue to round up until the end of mitosis. We also wanted to quantify the rising of the cells within the colony, and we did that by looking at the position of the cell center mass relative to the colony bottom here. And again, we see two nicely around the prometaphase stages that the colonies, that the dividing cell actually starts to rise up and then stays elevated throughout mitosis. The question arises, how can a cell undergo such a dramatic morphological change of rounding and rising up, but still being anchored within the colony? And one nice glimpse of an answer, I think, is shown in this beautiful movie where you have a plasma membrane gene-edited cell that is in a sea of unedited cells. And no, it's unfortunately not looping. I'll start it again. The top of the cell is in green, and the bottom of the cell is in purple. Sorry. Nope. So, yay. So on the top, you can see how more or less the top of the cell stays relatively localized, whereas the bottom of the cell like branches out and explores. And we see the similar trend when we look at the tight junctions. These are maximum intensity projections, so you kind of, it's like as you look from the tight junction down into the cells, and you can see as the cells start to divide, they maintain their tight junctions with their neighboring cells, but they're significantly ballooning up underneath those tight junctions. So this is a model how we're thinking how the cell shape changes as cells divide. They're going from their normal position within the colony, they're rising up, they're maintaining their junctions, but underneath their junctions, they're significantly rounding up. We can also look at how structures behave as cells divide. And I think this is one of the most beautiful slides I have ever shown in any talk that I have ever given. Those are our 14 structures. And we can do the classical approach. We can look at a structure over time in the cell cycle, and we can understand how the structure changes, how the morphology changes, where the localization is. But I think what is really not more interesting, but really exciting is that we actually can now look at all the structures at a distinct time in the cell cycle and try to integrate this to try to understand the organization of the cell at a specific time in its cell cycle. Unfortunately, I don't have time to go into all the structures today, but I would like to point out that in the beginning of prometaphase, where we also saw the cells rounding and rising up, this is actually where we see the most dramatic changes in structural morphology and localization as well. Since we had to choose a few structures just to highlight today, we chose the theme of disappearing structures or partially disappearing structures. The first one is the nuclear envelope. We tag laminin B1, and in interface you can see how it forms this nice laminar nuclear envelope structure. The nuclear envelope partially disassembles in the early prometaphase, cell cycle stage 3. and then reassembles as the cells undergo telophase and cytokinesis. And using live cell time lapses, we can actually use the nuclear envelope, lamin B1, to even divide our interphase stage into 3 stages of an early interphase stage where we see lamin B1 dots inside the nucleus and outside the nucleus in the cell, then a mid-interphase, nicely round nuclear envelope, and a late interphase one where we start to see invaginations in the nuclear lamina. Another structure that is studied that is disappeared is the Golgi. In our cells, if this is the nucleus, the Golgi kind of comes up as a tubular structure, and then we have a beautiful laminar network on top in interphase. As the cell starts to divide, the laminar structure on top fragments more. And then during the early prometaphase, state 3, the Golgi totally disassembles. We do not see the Golgi until it starts to reform at the last state of telophase and cytokinesis. Another beautiful structure is also fibrillarin. In the nucleolus, in interphase, we have a foci-looking structure. And then as the cells start to divide, what is actually really interesting in the early prometaphase, we see a phase transition, and we get from a more foci-looking structure to a melted structure. And then this melted structure totally disappears. And at anaphase and telophase and cytokinesis, we can actually see fibrillarin reassemble, but on the sister chromosomes. Before in interphase, they're again a structure of the nucleolus. So this is to end, is kind of to show all 14 structures again. If you have any questions or if you want to see any of the other structures, we have a poster. It's poster 20. Also, Dave in the team talk after the break will tell you more about how we want to integrate those 14 structures to understand the organization of the cell. And the Allen Institute is heavily based on teamwork, and not a single slide would have been possible without the hard work of all the teams at the Allen Institute. Thank you for your attention.